So today I will be talking about a study that we did looking at using comparative population genomics to reveal patterns of migration and structure in reef squid. When we think about the ocean's a very fluid and open environment inhabited by mostly very mobile organisms, which raises the question, where are these barriers to gene flow? It doesn't seem like there are very intuitive or obvious barriers that exist, raising the question, how do populations form and how does differentiation occur? And this has remained quite a long-standing question in studying speciation and population structure in marine ecosystems. So most marine organisms have a plagiochlorous stage. So even if they're benthic or sessile as adults, there's still potential for very wide distribution over a large geographic distance on ocean currents. So the long-standing assumption is that the duration of the larval stage, so how long an organism spends in the water column, is a pretty good indicator of how far they can disperse, and hence a good predictor of the degree of connectivity or the exchange of genes in individuals over geographic space. So under this paradigm, we must need very strong allopatric barriers to prevent gene flow. So a really interesting place to look at this is the Coral Triangle. Now this is a 6 million square kilometer region. It's bounded by the Philippines, Indonesia, Malaysia, and Papua New Guinea. And this is the center of marine biodiversity. It houses 76% of global coral species, 37% of reef fish species, and over 5,000 vertebrate species. So this is indicated by this, oh crap, sorry. By this red region here. I knew this happens at least once here. And there have been a lot of theories that have been regarding the processes driving such high diversity in this region. And what people have found is there are a few common genetic breaks that arise for most of the studies looking at or examining this region. And one such region is the Sunda Shelf. So this area was repeatedly exposed during the Pleistocene glaciation cycles, creating a land barrier to prevent gene flow between the Pacific and the Indian Oceans. Additionally, and that's indicated here by this line here, we also see a lot of patterns of population differentiation over different strong current features in the area, including here the Halmahera eddy, and then also here in the Java Sea, where there's a very strong seasonal reversing current preventing water flow between the north and the south. However, the majority of these studies inferring population structure and barriers to gene flow in the Coral Triangle are from benthic and strongly reef-associated organisms, organisms that do not move that far from their home reef in their entire life cycle. Therefore, our picture of connectivity in the region is incomplete, as we only really understand this from a particular type of life history. So then we must ask, do these same barriers to dispersal exist for highly mobile marine organisms? So as well, most of these studies infer these patterns from studies or from single or multi-locus studies using just a few markers. And this only indicates the patterns of connectivity. They're relevant over the time scale of these markers. So for most of these single marker studies, which are mostly mitochondrial CO1, this is a very long evolutionary time period that we're actually looking at. So what we're interested in is, do these processes still shape patterns of gene flow and dispersal in today's modern ocean? So in terrestrial systems, researchers have been more readily able to physically track or observe the movement of individuals. They can use tags and marker capture, whereas in the marine realm, we're limited. We can only spend so much time underwater observing where organisms go. However, with the advent of next generation sequencing technology, we can generate thousands of markers throughout the genome, which significantly increases the ability and the power available to assess changes in populations over increasingly ecological timescales. So for our study, we aim to look at these questions using a highly mobile marine organism, the big fin reef squid, Cichlidothis thysaniana, using a comparative population genomic approach. So these big fin reef squid are capable of moving over hundreds to thousands of kilometers, but they have no true larval stage. So they're something called paralarvae. So they develop over time in the water column, and when they're born, they're very strong swimming, because they can navigate readily through the water and spend a lot of time subjected to these potential dispersal barriers. They are neritic, meaning they're restricted to the continental shelf, but they do utilize shallow water habitats close to the coast for spawning activations. And they are very widely distributed. They are found throughout the Pacific and Indian Oceans, the Red Sea, and have recently, in the past few decades, have been found to invade the Mediterranean. However, within this nearly circumtropical range, our previous work has found that CP2 thistlesalliana isn't actually one species, that there's strong evidence for reciprocally monophyletic cryptic lineages within this group. And these cryptic lineages are co-distributed throughout the coral triangles. We find them in almost every single place that we've sampled. So for our study, we looked at lineages B and C, and having them being co-distributed so closely related throughout this range makes them a great system in which to test these shared barriers to gene flow. So from our main collection, we subsampled individuals from these different locations. So the green represents lineage B, and the purple represents lineage C, to test some of these known barriers to gene flow that other studies have found. So our samples are obtained from fish markets and from diving and snorkeling on spawning beds to collect the tissue samples. So we generated genome-wide SNPs from 1/16 of the genome using the 2B RAD method, which is a slightly different RAD technique designed by Wang et al. And it differs only in that it cuts 35 base pairs from the restriction recognition site, meaning that we have a higher likelihood that these fragments are going to be amplified for every single individual, minimizing the need to generate another SNP panel or assay to get all the markers that we want. So we amplified these fragments from 4 different locations for lineage B and 8 locations for lineage C, getting about 150 million and 322 million raw reads, respectively. Then we filtered these SNPs through a de novo pipeline that Misha Matz from the University of Texas and Eli Meyer from the University of Oregon designed. So we looked at SNPs that were genotyped at a minimum of 20x coverage, genotyped in at least 50% of individuals, and had these new alleles found in at least 2 individuals. And then we thinned all of these SNPs so that we only had 1 SNP per tag to minimize the effects of linkage disequilibrium. Finally, we removed outliers using Bayescan to minimize the effect of some type of selection, and we ended up with about 2,000 SNPs per lineage for this study. We then probabilistically assigned individuals to potential populations using admixture. So we tested K values from 1 to 10, but we are just showing here 1 to 6. And we find that K equals 2 had the lowest cross-validation error estimate, indicating that we likely have 2 likely populations for lineage B. And this here, we have 2 here, individuals sampled from Aceh and from Nha Trang in Vietnam form 1, and individuals from Manado and Raja Ampat in Indonesia form another. And lineage B is characterized by pretty high global FST. And when we break this down, we see that this high FST originates from comparing between these 2 populations. Whereas when we look within the populations, FST is much lower. So we have much lower differentiation between individuals within a population. Then for lineage C, this is the other cryptic species that we looked at. We tested the same K through 10, 1 through 10, and we found that most likely K was 3, corresponding to 3 different populations, with Aceh and the Andaman Sea over here in the Indian Ocean, the Gulf of Thailand, and Donggala in central Indonesia forming 1 population, with the Ryukyu archipelago forming another in Japan, and with Raja Ampat here in eastern Indonesia falling out by itself. And in comparison, lineage C has quite low differentiation overall. And even when we break this down between populations, we see that between populations we have fairly low differentiation, even lower differentiation within those populations. So when we start to compare these different lineages to each other and to the known breaks in the area, we do see a fair amount of differing patterns. We see here that for lineage B, that we have a strong break over this Makassar Strait area, which we don't see in other studies in this region. And these, the summary of these breaks also is from Ken Carpenter's lab and our lab in previous studies. And then for lineage C, we see a strong break here, or a fairly strong break here between Japan and the rest of the Coral Triangle. And we also see one here in Halmahera, which corresponds to other sites, primarily ones found for mantis shrimps. What we do see that's quite interesting is, remember I talked about this exposed land barrier during the Pleistocene, and we see that we actually don't have that differentiation, that there is quite high connectivity between the Indian Ocean, the South China Sea, and parts of central Indonesia. So when we think back on this, we don't see these same patterns when we start looking at markers that can change more rapidly over time and reflect possible more recent changes. And the breaks that we see seem to correspond to major surface currents. So the Makassar Strait, this area, has a very strong current called the Indonesian through flow that carries water from the Pacific to the Indian Ocean. So this could represent a very strong current barrier that the squid cannot physically cross or choose not to cross. Whereas the barriers that we see for lineage C, in the Halmahera eddy, there's a strong current coming over the north side of Papua New Guinea called the New Guinea Coastal Current, and this retroflects back going east. So this prevents any major water flow going east to west. And then we have Japan here. Not only is it separated by a very large geographic distance and some very deep water, but Japan also represents the northern edge of their range for these species. And as well, we have the Kuroshio Current coming out that cuts off the Ryukyu Archipelago from the rest of the South China Sea here. But recall that we're looking at two sister co-distributed species. And what we see is we see these very different patterns indicating that life history probably plays a very major role in influencing the responses to putative dispersal barriers. Oh, and there. Okay. And lastly, why is this important? Why is it so important that we know about the population structure and how these different species are connected over this space? So not only is the Coral Triangle the center of marine biodiversity, but it's also the center of threatened species for marine organisms. And this is due to significant amounts of habitat destruction and pollution in these countries, as well as severe amounts of overfishing. And so for these squid, these are a major fishery species for every single country in the Coral Triangle and the countries that we sampled, but currently there's very, very little management. And any management that does exist in Japan and Thailand isn't spatial. It's just purely on catch data. What we do see is we do see a lot of spatial structure, therefore we need to take this into account when we think about conservation and management of this region. Thank you to you all for listening, to my collaborators and funders, and everyone who's helped in the field. I can take one question. No question? Yes. Yes.
Are those based on mitochondrial DNA alone?
So that's based on 2 mitochondrial genes and 2 nuclear genes, and they were simply monophyletic for all 4 of those.